Vitae Discipuli. This is Mix Martin with lessons 10 and 11. So lesson 10 is about differentiation. The question is, how do cells with the same genes perform different functions? And the objective is explain how gene expression leads to differentiation. First section is gene expression. Let's remind ourselves what a gene is. So it's a heritable factor that controls a specific characteristic consisting of a length of DNA occupying a particular position on a chromosome. So we could take something like hair color. This is a bit of an oversimplification because there are multiple genes involved in hair color. But hair color is heritable, meaning it's passed on from parents to children. And the genes that control hair color we could find at a specific place on a particular chromosome. So each particular gene has an address. It has a place that it's found on a chromosome. And a gene is simply a length of DNA that codes for something. Gene expression, I'm not going to spend too much time on this because we covered it in the metabolism unit, but it's the process by which instructions in DNA are converted into a functional product such as a protein. So we start with DNA. Through the process of transcription, DNA gives instructions for the production of mRNA. Through the process of translation, mRNA gives instructions for making a protein. So remember that transcription happens in the nucleus, translation happens at a ribosome in the cytoplasm, so outside of the nucleus. In the process of transcription, DNA is used to create a complementary mRNA strand. So here in purple, we can see the DNA double helix. In the process of transcription, those two strands are separated and complementary bases are added. So G goes with C, C goes with G, C goes with G, A goes with U, because there's no T in RNA. T goes with A, and so on and so on. In translation, that sequence of mRNA is read by the ribosome in triplets of bases called codons. So every 3 bases comprises 1 codon, and every 1 codon specifies 1 particular amino acid. So here's codon number 1, and that specifies the amino acid arginine. Codon number 2, which is the second triplet, specifies the amino acid tyrosine, and so on and so on. Going on to differentiation. Think about this question for a second. Do all of your cells have the same DNA? So if I looked at a muscle cell, if I looked at a skin cell, if I looked at a brain cell, would they all have the same DNA? Think about it for a second. I hope you remember that the answer is yes. So the question that leads us to is, if all your cells have the same DNA, how can they have different functions? So how can your brain cell have a different function than their skin cell? How can your skin cell be different than a muscle cell? How is that possible? And the answer is differentiation. So differentiation is a process by which a cell becomes specialized for a specific function. So here we have a group of cells differentiating into muscle cells, blood cells, liver cells, and so on. Differentiation involves the expression of some genes and not others in a cell's genome. So even though all cells have all the genes in your genome, that doesn't mean that they're producing every possible protein at any moment. It means that they are expressing some regions of the DNA and silencing some regions of the DNA. It's like if you have a cookbook and you want to make a particular meal, maybe you'll look up 2 or 3 recipes. It doesn't mean that you make every single recipe in the cookbook. It means that you will select certain recipes based on what you want. And cells are similar. So here we have a neuron. It is a neuron because within the cell that became a neuron, the neuron-specific genes were expressed, meaning their proteins were made. Within the cell that became an epithelial cell, so that's like the lining of something is an epithelial cell, like the lining of your lungs, the lining of your small intestine. that became an epithelial cell because specific genes were expressed. So specific proteins were made, causing the production of an epithelial cell. This is what I was just saying. So if there are different expressed genes, we're going to end up with different protein combinations and different cell properties and functions. Take a minute to look at these questions. We're comparing a retinal cell, which is a cell in the back of your eye, to a red blood cell. So look at these 4 questions, pause the video, and try to answer. So for 1 and 2, you would find both genes in both cells because all of your cells have the same DNA. So you would find the gene for hemoglobin in both the retinal cell and the red blood cell. You'd find the gene for rhodopsin in both the retinal cell and the red blood cell. Remember that hemoglobin is the protein that carries oxygen within red blood cells, and rhodopsin is a protein that changes shape based on light. It's a light-sensitive protein that allows you to see. So it's found, it's part of your visual system. Number 3 says, in which cell would you find the hemoglobin protein? So you're going to find that gene expressed in the red blood cell, but not in the retinal cell. Because the retinal cell has no use for hemoglobin, it's not transporting oxygen, it's just doing its job helping you see. Number 4, in which cells would you find rhodopsin protein? You're not going to find that in the red blood cell, but you will find it in the retinal cell because it's part of making you be able to see. So the red blood cell has something to do with that. So you'll find the gene in both, but the protein only in the relevant cell.